Hello everyone, and welcome to this podcast for cell biology. In this podcast and the remaining podcasts of the semester, we're going to talk about cell communities, how they're established through tissues, and how these tissues are maintained, and that's what we're going to talk about today. And then later we'll talk about stem cells, and how stem cells are used to help replace dead cells in the tissue. And then we'll close with cancer, and how those cells within a tissue, how they can be transformed from a healthy normal cell to a transformed cancerous cell. But we're going to focus today on tissues and the cells within the tissues. Alright, so let's begin our discussion of tissues. Up to this point in the class, we've talked mainly about cells. We have talked about how cells communicate with other cells to some extent, but for the most part, we've talked about things that happen within a cell, and how the plasma membrane that surrounds the cell helps delineate this cell from other cells. And we want to talk now about how these cells can form communities of cells that bind together. Now, if these cells were bricks, it would be somewhat easy to understand how we could take these bricks and make a house from these bricks, because these are solid structures, very well defined. It's less simple to understand how you could take a single cell and make a tissue, and then how these tissues can be organized in such a way to make an organ. Let's say that's my stomach. It's not the best stomach, but you get the point. You make an organ, and then how those organs can be connected with each other through these interactions to form organ systems, and ultimately to form the organism. And once you move beyond this cell to these tissues, to organs, organ systems, and organisms, all the cells begin to take on different shapes and different structures, and the ability to communicate with each other and hold on to each other becomes critical. So it's much more complex than putting bricks together. No offense to people who build houses. We're going to focus on tissues. How these organ systems work within the organism, all that's going to come when you take a physiology course. But we're going to focus on tissues. And when we think about tissues, there are four kinds. We have connective tissue. This includes bones, cartilage, blood. We have epithelial tissue. There's nervous tissue and muscular tissue. We're only going to talk about these first two, because they help us understand two important parts of cell biology, which are critical to understand how tissues work and ultimately how organs and organ systems work. Connective tissues, we're going to have to understand the extracellular matrix. And we're going to talk about this in just a moment. Epithelial is a good way to introduce cellular junctions. How cells are held together, how they communicate between the different cells. This is going to be important. So we're going to talk now about connective tissue and the extracellular matrix, which I'm just going to abbreviate as EM. As we get started with our discussion of tissues, I think it's probably a good idea to define tissue. Just a general word. We can think of any of the tissues, any of the four tissues, as a cooperative assembly of cells and the extracellular matrix to form a distinct multicellular structure with a specific function. Since we're talking about connective tissues in this section, let's define connective tissue. Connective tissue includes bones, cartilage, the dermis, the part of our skin below the epidermis, and blood. Connective tissues are defined largely by their extracellular matrix. Since the connective tissues are largely defined by the extracellular matrix, it's important now to define the extracellular matrix. A lot of definitions here. The extracellular matrix is a network of polysaccharides and proteins that are secreted from cells. This extracellular matrix is a part of the tissue. I said that up here when I defined tissue, but I think it's important to remind ourselves that the extracellular matrix is not a substance distinct from the tissue. It is a part of the tissue. So the function of the extracellular matrix. It's important in protection. We can think of it as a spacer or a filler in between cells. It's very important in structure. In the role of structure, we can think of plants, and this extracellular matrix is what makes up the cell wall. In insects, this makes up the exoskeleton. In vertebrates, it makes up bones and cartilage. And all of this extracellular matrix, as I mentioned in the definition here, are substances secreted from the cell that provides these functions for that tissue. All right, so let's talk about some of the proteins involved in the extracellular matrix that's important in forming the connective tissues of things like bones, cartilage, and the skin. One of the major proteins found in the extracellular matrix is this protein called collagen. It is a very abundant protein. It is about 25% of all protein in mammals. All right, so collagen is made up of 3 individual polypeptides. Each one is a collagen protein, but they form this triple helix. This triple helix can then combine with other triple helices of collagen to form a larger structure called the collagen fiber. Several collagen fibers can come together to form the collagen fiber. Now that we formed the fiber, how does the fiber define the connective tissue? Meaning, how are we going to use these different fibers to create the various different kinds of connective tissues we see? Bone, cartilage, tendons, they're all very different. They have different elasticities, they have different strengths to them. This is accomplished in a few ways. One of the biggest ways is that there are at least 28 different collagen genes. And in different combinations, they will be found in different connective tissues. There will be a different combination of collagen genes in bone versus cartilage versus tendons. Also, the degree of packing. And what I mean by this is in connective tissues, sometimes the collagen folded all the way into a fiber, sometimes into a fibril. So different degrees of packing will lead to different strengths of the collagen being formed in that connective tissue. And the last thing I'm going to mention is that there are other proteins associated with collagen, depending upon the tissue type you're talking about. Now I want to talk about collagen-related diseases. On the previous board, I said there were 28 collagen genes, at least. Of the different collagen genes, the ones that are most predominant in mammalian cells are type 1, type 2, type 3, type 5, and type 11. The first 3 types are associated with diseases, and these diseases have fairly different phenotypes. So in the first one, collagen type 1 mutations will lead to a disease called osteogenesis imperfecta. This is characterized by very brittle bones. Individuals with this disease will have to be very careful because their bones will break very easily. This is particularly problematic during childhood as the bones are forming, and also problematic because if the disease hasn't been diagnosed, often parents are accused of child abuse. All right, collagen type 2 mutations. This leads to a condition called chondrodysplasia. This results in bone and joint deformities. And the last one to talk about is collagen type 3 mutations. This leads to a condition called Ehlers-Danlos syndrome. Individuals with this syndrome have fragile skin and also very stretchy skin. If you want, look on YouTube and for some videos of people with Ehlers-Danlos syndrome, and you can see how these individuals stretch their skin with great ease and in great distances. They also will have fragile blood vessels, which is very dangerous because this can lead to some circulatory problems. They also have problems with hypermobile joints. People with this syndrome I've seen have their joints often become dislocated, and so they exercise quite a bit to stretch out the joints, and it seems to prevent some of this dislocation that seems to happen a lot with people with this syndrome. So next I want to talk about the role of fibronectin, integrin, and actin in connective tissues. So how do these work together in connective tissues? So first, let me draw a plasma membrane. The integrin protein usually will exist in a dimer. And in this state that I've just drawn them, I'm trying to show you them as an inactive integrin. Integrins can bind to actin and fibronectin. Binding to either one of these will activate the integrin. And when it's activated, the two integrins will interact with each other. So this is an active integrin. And it's active because the actin cytoskeleton has bound to it. In addition, fibronectin has bound to it. So what we have here is the integrin, which is a transmembrane protein that is found both in the extracellular matrix area and the cytoplasm of the cell. You have the actin, which is in the cytoplasm, and the fibronectin, which is exclusively in the extracellular matrix. And what the fibronectin will do is it will interact with collagen. In this way, there's this mechanical interaction between the cell and the extracellular matrix, the collagen in this case. So I've explained to you how this complex forms and how it allows for this connection and communication between the cell and the extracellular matrix. But why is it important? Well, it's important for a few reasons. One of the reasons is it helps maintain cell shape. Another reason is that it helps the cell crawl or just move along the extracellular matrix. The plasma membrane here is full of these interactions that's attached to the collagen and other extracellular matrix material. But what it'll do is it'll continuously bind to it and release, bind to it and release. And when it does so in a coordinated manner, it will allow the cell to move with the extracellular matrix. They also, depending on the connective tissue we're talking about, help influence the collagen organization. Different organization of the collagen fibers will help determine the relative strength of that tissue and the type of connective tissue that's present. And a kind of a very specific note here, the actin holds the integrins to the cell. Without the actin attachment, as it's being moved and aligned with the collagen, these integrins would be pulled out of the cell. All right, that ends our discussion of connective tissue and the extracellular matrix. I'd like to move on to epithelial cells and cellular junctions. Epithelial cells are a very prominent cell in our body. Of the 200 plus cell types in vertebrates, most are epithelial. Epithelial cells cover our body as they are the epidermis of our skin, the outer covering of our skin. They also line our internal cavities. So our blood vessels, our respiratory system, our digestive system, any of our cavities are lined with epithelial cells. So these epithelial cells, they must bind with each other in a way similar to this. They can take on many different shapes and sizes, but for now I'm just going to draw them as this single layer of cells. So cells within the epithelium must, as I just said, bind to each other. They facilitate communication. So in the way they bind, they communicate with each other and between one side and the other side of the tissue, and they will allow for the passage of some molecules across the epithelium, but not all. So the way they're held together and the way they communicate is all going to be dependent upon cell junctions. So all of this is linked to cell junctions. So I'm going to draw the cells of the small intestines here, just to demonstrate and explain some of these junctions. So we have one cell here, and it's going to be attached to the basal lamina, a structure outside of the cell. And it's also going to be connected with a neighboring small intestine cell. We've drawn something similar before when we were talking about the transport of glucose from the lumen of the gut to the other side of the cell into blood vessels. So this is kind of like an old friend of yours just coming back from the past. The first one I want to talk about are tight junctions. And tight junctions, I'm going to draw here. Within the epithelium, what they will do is they will prevent the movement of fluid and molecules in between the cells. So they will seal cells together, blocking lateral transfer of fluids and other molecules between the two cells. These tight junctions that span from one cell to the other are made up of two proteins. One's called Collagen, and the other is called occludin. And that's all I want you to know about tight junctions. The next several I'm going to talk about together. They include the adherence desmosomes and the hemidesmosomes. All three of them are important in providing mechanical strength. So I'll write that one down here. All three in providing mechanical strength. They hold the cells together in a way that's different than the tight junctions. The tight junctions' whole purpose is just to prevent movement between the two cells. These other junctions hold the cells tightly together. The hemidesmosomes and the desmosomes interact with the cytoskeleton, and more specifically with intermediate filaments. And in doing so, that helps provide this mechanical strength. We call these desmosomes because they interact with desmosome proteins on each cell. These are hemidesmosomes because they involve an interaction between one cell and the basal lamina and not another cell. The adherents bind to actin filaments. I want to focus in on this adherence junction for a moment. So we have our two cells and we have our adherents. And the parts of the adherents include a linker protein that will bind to the actin and to a protein that will span the membrane and interact with a similar protein with the neighboring cell. And this is where the strength comes. And these proteins are called cadherents. All right, that's all I want you to know about adherents, desmosomes, and hemidesmosomes. The last one I want you to know are gap junctions. And these are my favorite. Strange twist of fate. Gap junctions are the reason I'm here today. Well, that is, here at CMU today. So let's talk about gap junctions. Gap junctions allow the transport of small molecules from one cell to the next. Molecules like calcium can move through here. It's a fairly small diameter, so you're not going to get large proteins going through, but very small molecules can go through. These are important in establishing that rhythm of muscle contraction for your heartbeat the rhythm and contraction necessary in the uterus during childbirth, some coordinated behavior that's necessary during embryogenesis. It's important in propagating a nerve signal. They're made up of 6 proteins, and these proteins are called connexins, one of my 2 favorite proteins. Together, these 6 connexins form this channel that allows the movement of small molecules. I would love to teach a whole semester just on gap junctions, but they won't allow me to do that. So that's all I'm going to say about them for now. And that's all I have for this podcast. If you have any questions at all, please let me know. If not, I'll see you in class. Bye.